Hello and welcome to today's lecture of Introduction to Mechanobiology. So, since the last two lectures I have started discussing of some of the tools and techniques that are useful for studying mechanobiology, and in the first lecture I introduced about hydrogels. Okay. What are hydrogels? What are their properties? Particularly what are some of the properties that you need to follow in order to make them in terms of tunability, okay, linearity of stiffness, etc. Okay. After hydrogels, I introduced AFM once more, but this time I introduced it how to measure properties of how to measure stiffness of samples with AFM, okay, and also to image. Okay. So, in AFM you have once again you could do protein unfolding as well. Okay. In AFM you could operate it in contact mode or tapping mode. These are two imaging modes. So, these are two imaging modes, contact mode being more suitable for flat rigid samples, okay, where there is no chance of damage to the sample, because you are in perpetual contact with the sample. Okay. And you have the tapping mode in which you are intermittent contact, and what you control is the amplitude set point. Okay. So, this is tapping mode is more suitable for imaging cells which are loosely adherent and can be dislodged upon lateral friction. Okay. And for measuring stiffness of samples of gels, cells, etc., okay, you use the force mode of operation. So, this is the same mode you also use for probing protein unfolding. Okay. And I had mentioned that in these, depending if your material is elastic, you get a curve which follows the indentation. So, the curve is indistinguishable between the indent and the retract zones. And if the material is viscoelastic like a cell, you get a curve like this, but there is one path of indentation and one part of retraction, suggestive of viscous damping. Okay. So, now one thing, when you measure the stiffness, okay, what has been observed is, let us say if you take skeletal muscle tubes, if you take skeletal myotubes, okay, and you measure their stiffness plus minus blebistatin. Okay. So, what you would observe is in the presence of blebistatin. So, if this is the so this is your stiffness axis and this is your frequency axis. Okay, this is your output data. If this is how your stiffness distribution looks in control or untreated cells, in blebistatin treated cells, the curve will shift to the left, or in other words the cells will appear softer. So, bleb treated cells appeared softer. Okay. But how do we think about it? Why is blebistatin, which inhibits myosin 2 activity, lead to a softer cortex? And the reason is as follows. Okay. So, if you think the cell as a tense string, okay, which is anchored at 2 points on a substrate. Okay. So, what you will see, so the string has its own material properties, okay, on top of which you are tensing the string, you are adding some tension in the string. Okay. So, what is the source of this tension? If you anchor it to the substrate, the source of this tension is actomyosin contractility. Okay. So, if you inhibit the tension, so in essence if I relate the tension to the overall stiffness that you measure, this should be a straight line. Okay. So, greater amount of tension you impose. So, if you pull the string, then if you try to deform it, you need extra force to deform it. In other words, the string appears to be stiffer. Okay. So, this tells you that cell stiffness depends on contractility. Okay. So, this suggests that in a sense you can get an idea of how contractile a cell is by measuring its stiffness. However, you will not see the effect of contractility on stiffness if your cortical contractility does not change, is unchanged, but you have upregulation of rho or rock such that there is stress fibers which are built up in the center of the cell. Why? Because if this is your nucleus, you are probing from the top. So, if you have stress fibers which are right next to the nucleus, you cannot, these won't contribute as much to the overall stiffness that you are probing. So, there has to be a different way of probing contractility independent of measuring stiffness. Okay? And one of the most well established ways of probing contractility is traction force microscopy. Okay. So, let me explain what exactly is TFM. Okay. So, if you have a cell sitting on a substrate of given stiffness E, okay, and what I can do is, let us take a section of the cell here. So, what you have is a section of the cell, okay, the cell is exerting internal forces. Okay. This is the source of your pre-stress P, and these forces are stabilized by tractions at the substrate at the interface of cells and gels transmitted to the focal adhesions. Okay. So, this is your T bar. Okay. So, you must have a force balance required by P. So, if I were to draw the whole cell in this way, this is a 3-dimensional surface, then P into this cross sectional area, let us say A bar, must be equal to T, okay, dot this base area AB, A base. Okay. This is the force balance, and this is the basic ideology of doing traction force microscopy. But how do you measure these tractions or these forces on the substrate? Okay. What is typically done is, in the PA gels or PDMS gels that you fabricate, okay, you put some fluorescent beads on the surface. Okay. So, you might, and how do you get these beads on the surface? You polymerize, okay, you polymerize the PA gel, polyacrylamide gel, with beads inside. Okay. So, there are of course lot of beads in other planes as well, but you are more focused, and you polymerize this gel upside down. Okay. So, if you polymerize the gel upside down, then because of gravity, all these beads will remain at the top surface of the gel. Okay. So, and then, so if I were to draw the cell in 2D, and this is your substrate. So, you have these beads sitting in and around the cell on the top surface. Okay. So, if there were individual focal adhesions through which the cell is exerting forces inside, if let us say these are 4 focal adhesions through which cells are exerting forces in the given direction. So, what you do after you plate the cells on these substrates, okay, you take one image in the stressed configuration. Okay. So, imagine these black dots are the positions of the beads in the stressed configuration. Then what I do? I remove the cells using trypsin. Okay. So, if these were the original directions in which the cell was exerting forces, once I remove the cells, these beads would move in the outward directions. But the beads which are far from the cell, so these beads may not have effective any displacement, but these beads which are positioned close to the cell will move in outward directions. Okay. So, from this what I can do is I can track what is the bead displacements. Between stressed and unstressed configurations. Okay. So, unstressed corresponds to the configuration when the cell has been removed, and stressed corresponds to the configuration in which the cell is actively exerting forces on the cell. So, using this, once you get the bead displacement, then you can use theory of linear elasticity to find out what is the force distribution. Okay. And these experiments have been done for multiple different cell types, as a consequence of which, say for example, if this is a muscle cell, you would find broadly 2 big forces which are equal and opposite in direction, but the points of exertion of these forces are separated by a distance d. Okay. So, this is for a muscle cell. So, this corresponds to what is called as a force dipole. So, this is an example in which you can exert contractile forces along one direction. This is applicable for cells which are elongated like fibroblasts or muscle cells. Okay. If you were doing the same experiment for your osteoblast, so which is a more, the morphology of an osteoblast is more polygonal in nature, and if you do the force distribution, you would see the force distribution as radially symmetric. Okay, radially symmetric. And using these experiments, lot of people have shown that again that there is a linear relationship between contractility and stiffness. Okay, you should have a straight linear relationship. So, this means that if you plate, so one more observation has been for gamut of cell lines. If you do with stiffness, substrate stiffness, you always see contractility increasing. Okay, contractility exhibits increase with stiffness. Okay. So, this approach is very powerful, but in this particular type of traction force microscopy that we discussed, this is called the 2D traction force microscopy. So, what it assumes is when you are having a cell on a substrate, the cells are exerting inward direction forces in the xy plane. So, this is your xy plane and this is your, so you have x, y and z. So, all the forces are being exerted by the cell in the xy plane, which is not necessarily true. So, here you do not account for bead displacements which are in the z direction. Okay. So, instead of pulling like this, you could very well imagine the cell exerting a force like this, In that case, there will be z displacement of the beads as well. Okay. So, this is what is taken into account in 3D traction force microscopy, where cells are embedded in gels and you have beads in around the cell in all different directions, and you track the x, y, z movement of the beads. So, this is of course way more complicated, and it requires important particle tracking algorithms to find out, to detect the position of individual beads and to track their displacement. Okay. So, one of the caveats or challenges with traction force microscopy is, imagine how can you use traction force microscopy if cells are cultured on glass. Okay. Or let us say on cell derived matrices. Okay. So, where there is no way of adding beads into the material. Or one more example is if your material is non-linearly elastic, because the formulation for traction force microscopy assumes linear elastic substrates. Okay. For all these cases, so there is a very simple alternative approach which you might follow. This is called a Trypson deadhesion assay. Okay. So, what is the Trypson deadhesion assay? So, this is what you do routinely, right. So, in culture you use trypsin for passaging. Okay. So, if you have a cell which is adherent and you add trypsin, okay, then at some point of time it should round up. Okay, and in between you would have a range of different configurations where the extent of rounding up is increasing. So, cell goes from a completely spread configuration to a round configuration. So, you can actually track the dynamics. So, what if you take a time lapse image, time lapse movie of a cell rounding up, when incubated with trypsin, okay. So, what you will have is a plot of time and area A. Okay. At t equal to 0, the initial area of the cell is A, at some time t it is some AT, and finally at some time Tf you have a final area of AF. Okay. So, I can define this normalized change of area defined as okay, I can define this normalized quantity A bar as Ai minus A of time t by Ai minus A of f. So, if I do this, then and if I plot A bar at t equal to 0, A of t is same as Ai. So, you have a value which goes from 0 all the way to 1. So, if you plot A bar as a function of time, you would have a curve. Okay. Your A bar curves look something like this, and what has been observed is this curve is sigmoidal in nature. Okay. So, for a sigmoidal curve, what you can do, you can fit this data by the following expression. Okay. I can have this particular function A bar fitted to this expression, where I can find out two time constants tau 1 and tau 2 is. So, what is tau 1? Let us see, at t equal to tau 1, this entire variable becomes e to the power 0. So, that is 2. So, 1 minus half becomes half. Okay. So, tau 1 corresponds to the time at which you have reached a value of A bar equal to half, and tau 2 is the time constant for the second portion of the curve. Okay. So, what has been observed? So, you can backtrack two time constants tau 1 and tau 2. Okay. What has been observed is when you perturb cells with contractility inhibitors, okay. So, what you see, let us say if this is the value of tau 1, let us say this is my tau 1 and this is my tau 2. For control cells, let us say this is my value of tau 1 and tau 2. When I treat them with drugs like blebistatin, okay, like bleb, which inhibits contractility, both these time constants tend to increase. Again, when I treat with drugs which promote contractility, well, let us say lysophosphatidic acid, which promotes the Rodock pathway, then what you see is your tau to tau values decrease. In other words, de-addition time scales are dictated by contractility. Okay. So, if a cell is more contractile in nature, then when you treat with trypsin, it will round up that much faster. Okay. So, what has been also observed is by correlating these time constants with the value of stiffness using AFM, what is observed is tau is generally inversely correlated with that of stiffness. So, for a cell which is stiffer and more contractile, its de-addition time scale will be lower. Okay. So, this is a very simple assay based on which you can estimate how much is the contractility. The caveat is, so one of the caveats is you are estimating contractility indirectly in terms of time constants and not in terms of actual amount of forces exerted by the cells. The other caveat is, okay, that tau is not only dictated by contractility, okay, it is also dictated by additivity. Okay. In other words, imagine I have two conditions in which contractility is constant, C is constant, but additivity changes from A1 to A2, where A2 is significantly greater than A1. Okay. So, what you will observe is if this is the profile, de-addition profile for a given cell with A1, as you make it A2, so what you will see is this de-addition will be delayed. Okay. So, this is for value of A1, this is for value of A2. So, in other words, the de-addition response is not only dictated by contractility, but also dictated by additivity. Now, you can perturb the additivity might change because of overexpression of vinculin, let us say, of a focal adhesion protein like vinculin, which is a structural protein. Okay. But you can also change this by adding trypsin, which is 10 times more active. Okay. In that ways, the chopping rate of focal adhesions is going to be higher. Okay. So, compared to traction force, so both TFM, okay, and this de-addition assay, they kind of estimate the whole cell level contractility. Okay. Is it possible to estimate the contractility in a single stress fiber? Okay. So, in other words, what is the contribution of a single stress fiber on the overall cell contractility? Okay. So, in these cases it is difficult to use TFM or de-addition assay to estimate. Okay. So, there is a new technique which has been introduced, it is called laser ablation. So, imagine you have a cell which is transfected with, where you have transfected cells with life act, and you can have very prominent stress fibers. So, these are very prominent stress fibers. So, what laser ablation does is it focuses laser light at a single point on a stress fiber. So, when you focus laser light on a single point on a stress fiber, the energy is so high that locally the material just evaporates. So, using femtosecond lasers, you focus light onto a nanometer sized intracellular target. So, this light is so focused, this will lead, so this will lead to evaporation of the sample. At a single point, at the point where you have focused. So, what you will have, so imagine I have this cell and this is one such stress fiber in which I had focused light at this single point. So, and you acquire the fluorescence images as a function of time. So, what you will see is this stress fiber, it will relax. So, both these ends A and B, so you have locally disrupted this stress fiber. This point will retract along the line backward and this point will retract along this line in this direction. So, as a consequence, this length, if I actually track the gap G as a function of time, what has been observed is this G as a function of time exhibits a saturation profile like this. So, there is an instantaneous increase in gap and then it slows down to some eventual value. So, just like previous case, you can fit this data with let us say dA plus LA e to the power minus t by tau, you can get an estimate of the characteristic time constant of relaxation and also you can estimate this LA value. So, what has been observed is when you treat cells with contractile agonists, if this was the response for a control cell, when you treat with Y27632, it drops. With blebistatin also it drops significantly. So, this suggests that this is also an example of how contractility at the single stress fiber is being modulated. Using this experiment, Sanjay Kumar demonstrated that when a cell is sitting on a compliant substrate, when you have a single stress fiber being ablated, as a consequence of this single stress fiber, because the substrate acts as an elastic link for two different points within the cell. So, when you even if you ablate a single stress fiber, there is global rearrangement in the cell as a consequence of the single ablation, but this is markedly absent if the cell was connected in a or cultured on a glass substrate, suggesting that transmission of forces. So, this experiment suggests that transmission of forces through substrate regulates the force balance in a single cell. So, this regulates your tensional homeostasis or the force balance. So, just to summarize, today I have discussed three different techniques: traction force microscopy, de-addition and laser ablation, all of which provide some estimate of contractility. So, de-addition provides the cell level contractility, TFM provides contractility information within the cell at different points, and laser ablation also provides contractility, but from a single stress fiber. Okay. So, both these two techniques, okay, will provide this information in the form of time. Okay. Lesser time is correlated with higher contractility, while in TFM you get actual estimate of forces exerted at different points within the cell. Okay. With that, I thank you for your attention, and we will continue in next class with two other techniques. One is microfabrication and the other is fret. Thank you.